The overall goal of the following procedure is to evaluate the innate sensing of HIV-1 infected CD4 positive T cells by plasmacytoid dendritic cells, or pDCs. This is accomplished first by infecting CD4 positive T cells with HIV-1. In the second step, the infected T cells are co-cultured with pDCs. After 18 to 22 hours, the supernatants from the co-cultures are recovered for quantification of pDC type 1 interferon production. Ultimately, the production of type 1 interferon can be used to assess the sensitivity of human blood-isolated pDC to CD4 positive T cell HIV-1 infection.
This method could be useful for studying viral and host factor governing the early innate immune response in recognition of pathogens, such as the recognition of HIV-1 infected CD4 T cell by plasmacytoid dendritic cells.
Although these protocols are aimed at measuring type 1 interferon, they can be easily modified to detect other bioactive molecules released by pDCs upon recognitions of infected T cells, such as interferon gamma, TNF-alpha, or chemokines.
For the infection of primary CD4 positive T cells, begin by activating human peripheral blood isolated CD4 positive T cells with PHAL for 48 hours. Maintain the cells in complete medium supplemented with recombinant IL-2 for at least 3 days prior to infection. Then, in a biosafety level 3 laboratory, infect the T cells with HIV-1 viruses using different multiplicities of infection. 2 days post-infection, evaluate the cell cultures for their expression of GFP, BST2.